Good morning, good afternoon, and good evening to everyone joining around the world. Welcome to the Global COVID Lab Meeting. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. We've had a little break since our last meeting, and so much has happened. We've had new clinical trials with COVID vaccines have started at a record pace, and the volume of new data and insight being generated around SARS-CoV-2 is truly remarkable. In this recurring lab meeting, we're focused on bringing you the latest research pertaining to COVID-19 vaccine and therapeutic development from experts in a forum open to the global scientific community. To date, over 1,300 people have registered for the lab meeting, and we've received a lot of great feedback. So thank you for your interest and enthusiasm around the lab meetings. I'm very excited for today's speaker, Dr. Shane Crotty. Dr. Crotty is a professor at the Jolla Institute for Immunology and an adjunct professor at the UCSD School of Medicine in the Division of Infectious Diseases and Global Public Health. Dr. Crotty's lab studies the fundamental immunology underlying the functions of vaccines. His team focuses on both the basic immunology of T follicular helper CD4 T cells, as well as the central roles of germinal centers and memory in vaccine immunology. Dr. Crotty was named a Pew Scholar in Biomedical Sciences in 2005 and was the 2012 recipient of the American Association of Immunologists BD Biosciences Investigator Award for outstanding early career research contributions to the fields of immunology. And he is the Cancer Center, he was recently named a 2016, 2017, and 2018 Thomson Reuters ISI highly cited researcher, and he's the Cancer Research Institute's Frederick W. Alt Award for New Discoveries in Immunology. In today's lab meeting, Dr. Crotty is going to talk about T-cell responses to SARS-CoV-2. During the presentation, all attendees have the opportunity to send questions using the Q&A function in Zoom. Please use the Q&A function and not the chat so that I have a chance to see your questions. I'll ask Dr. Crotty a broad selection of these questions after he's finished his presentation. We'll have about 25 minutes for discussion, and I hope you'll take the opportunity to ask your questions about the data presented today. It is now my pleasure to introduce Dr. Shane Crotty from the Jolla Institute for Immunology.
All right, thanks everybody for joining. It's a pleasure to be here. Yeah, so for those who aren't familiar with the Jolla Institute, we're a nonprofit research institute on the University of California, San Diego campus. We essentially operate like a freestanding immunology department, and it's a fantastic place for all kinds of immunology, certainly infectious disease immunology. And I've been collaborating with the Human Vaccines Project since the beginning, actually. It's a fantastic organization. It's really been a pleasure to work with the Human Vaccines Project. And the COVID newsletter they've been putting out has really been fantastic. I've really found that to be some must-read articles about perspectives on COVID. So it's a pleasure to present today. For the many of you who aren't familiar with my lab and our work, I'm generally obsessed with vaccine immunology, and almost all licensed human vaccines work on the basis of protective antibody responses. And so my lab focuses on trying to understand the fundamental immunology underlying those protective antibody responses. What are the cells involved? What are the steps involved? How do you get to this red box over here, which are essentially the cells that are the key effectors for most human vaccines? And this animation summarizes the key differentiation steps of TFH cells. Well, and I'll back up just a smidge. So in this, so it's the B cells, of course, that make the antibodies. And I know the audience for these webinars is quite broad, going all the way from high-end experts to novices. And so I will try and give a bit of introduction. And these responses, these antibody responses, generally completely depend on CD4 T cells to help the B cells. And those CD4 T cells that provide the help are T follicular helper cells. That's the subset of CD4 T cells. And this is an animation of TFH cell differentiation and interaction with B cells. What we in many labs have learned over the past 10 years now are that TFH cell differentiation occurs quite early in an immune response, within the first few days frequently. And those TFH cells are the CD4s that are important for interacting with the B cells from early time points onward. So B cells, in many cases, can essentially undergo one cell division without T cell help, and all activities of B cells after that depend on the presence of CD4 T cells, and particularly TFH. And those TFH can not only drive the B cell proliferation or selection of the B cells against the viral infection, but also drive their differentiation into the plasma cells or plasmablasts, the actual antibody secreting cells. And that's important both early on, so that an initial wave of antibody is made to an ongoing infection. But really the most extraordinary part of antibody responses and T cell interaction with B cells comes in germinal centers, where in many cases the B cell response to the antigen is initially relatively low affinity. And so the germinal center is the place where the B cell response mutates and evolves into high affinity B cells capable of making high affinity antibodies, which are thus much more effective at neutralization, because potency of neutralization is associated with the affinity. And T cells play a central role in that. The TFH cells are actually required to select the B cells, drive their mutation and proliferation, and iterate those cycles until hopefully one ends up with high quality B cell memory, circulating antibody, and memory TFH cells as well. Okay. With the advent of the COVID pandemic, my lab and that of Alex Setty's lab pivoted to try and address key questions about immune responses to SARS-CoV-2 in the context of COVID-19, and that's what I'll focus on today, walking through what we recently published in this article, and talking about what's been happening since. Okay. And really, like any new infectious disease, there have been major knowledge gaps in understanding immunity to this new virus, SARS-CoV-2, and major questions have been how much of an adaptive immune response is there in COVID-19 disease. There were concerns that there were essentially no adaptive immune responses, or highly aberrant immune responses. Another huge question is how long does immunological memory to COVID-19 last? And one can infer some aspects of this from how large the initial immune response is, although there's no substitute for actually measuring memory by having to wait, but you can at least get some sense early on from the initial magnitude. And then what kind of immunity is important against COVID-19? Now, each of these questions are important both from a disease standpoint. How does one end up with severe disease versus mild disease? Does that have to do with the nature of the immune response to this disease, or is it completely unrelated to the immunology? Is it related to non-immunological risk factors? And also, critically, not only does this relate to factors of ongoing disease, but also vaccine development. And really, for vaccine development, you really want to know, you would really like to know up front what kind of immunity is important for that vaccine naturally. So when people get infected, resolve that infection, what parts of the immune system are bringing about that resolution of infection, because those protective components of immunity are ones that you would likely want to try and engage in vaccine development. And of course, the COVID-19 pandemic has been more of a world on fire type situation, and so it's been important for people to go ahead and start vaccine development without having these answers. But as fast as we can develop these answers, they will hopefully be able to inform vaccine development about what may or may not be on the right path. And I'll comment on some of that as we go along. And that was really a big driver of what Alex Setti and I did. Do people develop immunity to COVID-19? Well, it is an acute infection that resolves in most humans, and so that is an indication that the adaptive immune system is probably doing something, and it is resolving the infection. And we try to develop direct evidence of that. And again, what type of immunity is important to it? There are three major arms of the adaptive immune system. Antibodies made by B cells are one of those arms. And as I mentioned, antibodies are important in almost all currently licensed human vaccines. And so, of course, for COVID-19 vaccine development, most efforts have focused on generating neutralizing antibodies against COVID-19, and that is a logical approach. However, it's not the only approach, and the effectiveness of such approaches does depend on the nature of the infectious disease. And so, for example, as there are, as I said, three arms of the adaptive immune system, there are three arms of the adaptive immune system for a reason. They're generally all three of them valuable in responses against viral infections. And the CD4 T cells are critical for antibody responses, so it's highly unlikely that you can get a neutralizing antibody response that's durable to COVID-19 without a good CD4 response. But separately, CD4s can do a wide range of functions, including antiviral activities. And to SARS-1, classic SARS, it was previously shown in mouse models that CD4 T cell responses could actually protect against a lethal SARS challenge in mice in the absence of antibodies or CD8s. And so, considering the roles of CD4s in protective immunity against COVID-19 makes a lot of sense. Last but not least, there are the CD8 T cells, right, which are the killer T cells. Antibodies are wonderful, but antibodies almost exclusively act on virus outside of infected cells. And CD8s do the opposite, right? They eliminate infected cells and have clearly been shown to be important against for protective immunity against many viral infections. And of note, there have been two individuals reported who are agamaglobulinemic adults who contracted COVID-19 and survived without hospitalization. Indicate these are people who have no B cells from birth, and so are incapable of making antibody responses, and yet they resolve the infection, indicating that at least in some cases, a T cell only response against this virus can result in resolution of the infection. Okay, so we set out to measure all three arms of the adaptive immune system to COVID-19. To do that, for T cell responses, this can be challenging because when you have a virus shown here in Lego form as SARS-CoV-2, T cells recognize peptide epitopes of the virus, and so they can come from any given part of the virus. And so it's key to identify those epitopes or predict those epitopes. And Alex Setti, faculty here at LJI, is really one of the top couple of experts in the world at predicting T cell epitopes and identifying antigen-specific T cells in humans. And so he and I collaborated together on this project, and we did so with three key concepts in mind. Okay, to us as a benchmark study, it was important to focus on average cases of COVID-19. Okay, so the majority of COVID-19 cases do not require hospitalization. They result in essentially flu-like symptoms or other related symptoms. And so we wanted to assess what occurred during those infections. I don't know how to turn that off. As a reference point. Second, since we did not know what the characteristics of the T cell response were, we felt it was important to take an agnostic approach to measuring those T cells, i.e., we didn't want to miss the T cell response because we were looking for the wrong cytokine. And I'll talk more about that in a moment. And then third, with a genome the size of SARS-CoV-2, it can be challenging to predict T cell epitopes and also know which parts of the virus are actually being recognized by a human T cell response. And so we felt it was important to take a dual approach for measuring T cell responses, both taking a response of predicting epitopes, which the SETI lab is very skilled at, but also take a full ORF scanning approach to make sure we weren't missing responses. Okay, and so I'll talk through all three of those approaches and their results. So first, we looked for the antibody responses in these patients, and so here we're focusing on 20 convalescent cases of average COVID-19 from the San Diego area, and we measured RBD, the receptor binding domain of SARS-CoV-2 spike, IgG responses, IgM responses, and IgA responses, and in general, all 20 individuals were positive. And for the T cell responses, as I mentioned, we wanted to take a cytokine agnostic approach so that we would not miss what type of T cell responses the infected individuals were making. And so in general, what we've done for this is we've made use of AIM assays, which are short for T cell receptor dependent activation induced marker assays. When T cells see antigen, they can make certain cytokines, but they also upregulate surface molecules. And they can upregulate a range of these surface molecules, CD40 ligand, OX40, 4-1BB, CD69. And so we and other labs use these as cytokine agnostic approaches. And we demonstrated that for TFH cells, the CD4s that help the B cells, this can be a critical approach actually to properly identify the cells, particularly in lymphoid tissue. When looking at germinal center TFH cells, these cells are actually very stingy cytokine producers. And so cytokine assays can miss 98% of the antigen-specific cells, which instead AIM assays can detect. And we've used this approach with the SETI lab to study the pertussis vaccine, where it was quite difficult to identify antigen-specific CD4 T cells against pertussis with conventional approaches. And by using AIM assays, they were detectable. And notably, since the AIM assays are live cell assays, we don't have to kill the cells, unlike intracellular cytokine stains. This experimental approach is then amenable to RNA-seq and downstream transcriptional analysis. And so, for example, in the pertussis case, we and the SETI lab could observe that these cells made cytokines that we didn't expect that they would be making. And we've also used this extensively in the context of HIV vaccine efforts, particularly in monkeys, rhesus monkeys, where, as an example shown here, immunized animals, when we looked for interferon gamma specific CD4 T cells, most of the animals were undetectable. But by AIM assay, we could see that actually all 12 animals participating in the study had made CD4 T cell responses specific to envelope, and those responses were equivalent. Okay, and we've applied this in humans, in infectious disease, and allergy and other contexts. So we did so for these 20 COVID-19 cases. And we did this with two major antigen targets, okay? And in one case, we took overlapping peptides against the entirety of spike, and we did this because spike is the target of almost all of the COVID-19 vaccine programs that are ongoing, right? I mean, at least lists I've seen, if people list 100 vaccines, something like 98 of those are spike-only vaccines. And so we considered it critical to measure, well, do people who actually have COVID-19 make CD4 T cell responses to spike, and what do those T cell responses look like? Because one possibility was that spike actually was a terrible CD4 T cell target, in which case that would be a red flag for vaccine strategies. However, we observed that spike is actually an excellent CD4 T cell target. We saw virtually all infected people made CD4 T cells specific to spike. And then separately, we had a non-spike pool. And so this is a pool of predicted class 2 epitopes made by the SETI lab, led by Albo Grifoni. And we could see here that their predictions were good, essentially, that this epitope pool, as a single pool, and thereby needing very few reagents, very few cells to run the assay, was actually effective at detecting CD4 T cell responses in most people. And then what we did was we took a combined metric. And here's example raw data over here. Okay. We did similar things with the CD8s. And so here I'll just present it as summary data. What we observed were, in average cases of COVID-19, we observed SARS-2 specific CD4 responses in 100% of the cases, IgG in 100%, IgA in 80% to spike, and CD8s in 70%. And overall, we consider that good news. Really, I certainly won't claim that it's perfect, but given the amount of fear about what might be going on, we consider all of this good news. Essentially, the viral infection was immunogenic in these cases, and in addition, the CD4 T cell responses, which I'm not showing here, looked like antiviral CD4 T cells. They were they predominantly made interferon gamma, which is what one would expect, and we didn't see much evidence of aberrant skewing of CD4s. And so frankly, if you told me that there was a new respiratory pathogen causing flu-like symptoms in people and asked, what type of immune response would I expect to see and of what magnitude, this would largely fit my de novo expectations and predictions. Okay, so it mostly looks as expected, again, in these average cases. We observed good responses to spike for CD4, and we also observed CD8 responses to spike. Again, that's of interest since spike is such a strong focus of vaccine programs. We again consider this good news. It looks to us like spike is a reasonable target for most vaccine programs, as it elicits good CD4 responses. But we also saw strong responses to other targets, which I'll get to more in a moment. Additionally, in terms of the qualitative features of the CD4 response, we wanted to know if it looked like those CD4s were providing good T cell help to the B cells. And so we looked at correlations between the spike-specific CD4 T cells in infected people and the IgG titers to RBD, and there was a good relationship between those two, okay, indicating that a reasonable fraction of the spike-specific CD4s probably were TFH cells and probably were providing good help to B cells. Again, largely antiviral immunity that matches expectations, and we again counted as good news. Okay, so that was the first part of the experiments, and Alex said he really knows what he's doing, and he knows it's important to be rigorous in initial characterizations of T cell responses to make sure when trying to map epitopes that you're not missing a big piece of the puzzle. And so he really committed to synthesizing overlapping peptides to the entire genome, which was a big and expensive effort, but we really felt was important for establishing a benchmark. And so when we looked at the CD4 T cell responses, actually in a new set of 10 donors here, we could see CD4 T cell responses to actually many different proteins of SARS-2. And at first glance, it was tough to interpret the nature of those responses. But then we realized if we reorganized the ORFs based on our prediction of what would be most expressed in infected cells, we saw a pretty clear picture. So SARS-CoV-2 as a coronavirus doesn't express proteins in equimolar ratios. It uses subgenomic RNAs to amplify expression of some proteins over than others. And actually at the time we were inferring protein abundance based on RNA abundance, well, actually based on just data from other coronaviruses. In data published since, that's confirmed almost all of our predictions here about the abundance. And indeed, the CD4 responses in COVID-19 infected people largely track with the abundance of those proteins in infected cells, and that M, N, and S are highly expressed proteins and end up being highly immunogenic for T cell responses. And interestingly, it's really quite a broad response. We identified responses to 21 out of 25 of the ORFs. Okay, and so that's summarized here. Really, again, good news in general. And so that's the summary of the immune responses from the infected individuals. Now, the second part of that project that became quite interesting was that we observed SARS-2 reactive T cells in unexposed normal healthy donors. And we checked this multiple different ways, and we could be confident of what we were seeing because we were using blood samples collected in 2015 to 2018. So before there was any SARS-CoV-2 around, these blood samples were collected. And when we looked, in these blood samples from these people, we saw that about 50% of the people had CD4 T cells that could already recognize SARS-2 and could in fact recognize several different ORFs within SARS-2. And there were also rare CD8s that could do so as well. And so from this, we concluded, well, we speculated that it seemed likely that these were memory CD4 and CD8 T cells that were coming from a person's previous exposure to common cold coronaviruses. And we don't know this hasn't been directly demonstrated, but we speculated this based on what's known about common cold coronaviruses. Okay, so there are other coronaviruses that infect humans. There are 4 what are generally referred to as common cold coronaviruses: OC43, 229E, NL63, and HKU1. That were identified at different points over the past 60 years. And these viruses circulate regularly in the US and elsewhere with different patterns. And so here's data from the CDC showing actually the overall circulation of these 4 common cold coronaviruses in the United States over the window from 2015 to 2017. And what you can see is that all 4 of them circulated, and all 4 circulated actually with different patterns. And so overall, that leads to, well, some very interesting speculative possibilities, which I'll discuss in the next slide. So to summarize, we've observed that in average cases of COVID-19, we see reasonable antiviral immune responses. 100% of people were making CD4s. A reasonable people had detectable CD8s, and they generally seroconverted. And that there were responses against spike as well as a number of other antigens. And so we in the SETI lab, in terms of what's next, have been working with vaccine developers about best practices for measuring CD4 and CD8 T cells in responses to vaccines, in terms of sharing epitope reagents or T cell assays, as well as making use of data from COVID-19 patients for as benchmarks, right, for what is a reasonable magnitude for vaccine-elicited CD4 or potentially CD8 T cell response. The SETI lab is very hard at work at fine-scale epitope mapping. What we reported were responses to pools of peptides. And so the next step is to map individual epitopes, and that work is ongoing. Third, we did this work as a benchmark, right, for what a good immune response to COVID-19 looks like, right? What an average protective immune response to COVID-19 looks like. But it's also very important to measure immune responses in hospitalized cases and in acute cases. And we're hard at work at that. There have been various reports and publications of aspects of this, but none showing actually antigen-specific CD4, CD8, and neutralizing antibody responses to date. So we are doing that to complement our previous work, and hopefully numerous other groups are as well, to get insights into are there differences in immune responses in people who have severe or hospitalized COVID-19 compared to people who have non-hospitalized cases. Are those immunological differences or other differences? Lastly, on this slide, we think these data do support that next generation vaccine designs should consider T cells. There's clearly broad CD4 and CD8 responses to this virus, and various groups have published that neutralizing antibody responses in COVID-19 infection are not particularly strong, and so our data suggests that there's a good chance that T cells are playing an important role in protective immunity here, and should be considered in vaccine designs. And finally, to wrap up and then shift to the Q&A, this pre-existing immunity we're seeing in people, these pre-existing SARS-2 reactive T cells, have a number of different implications. One thing we've been doing is highlighting this topic for vaccine developers, because we do think it's important to measure pre-existing SARS-2 reactive T cells at time 0 in vaccine clinical trials, because people, if it is like 50% of people who have such cells, that could result in substantial differences in outcomes between people who have those cells at time 0 and people who don't. And phase 1 clinical trials are quite small, so you could stochastically end up with unexpected results that just have to do with who you happen to enroll. And our observation of these pre-existing cells has now been recapitulated in 5 different studies, including on 3 different continents. So these pre-existing SARS-2 reactive T cells are real. But what isn't known is what are they? For example, it hasn't directly been demonstrated that they're memory cells. And 2, do they matter, right? And so there's been a lot of speculation on this topic, and we've speculated as well. But just to be clear, we don't have any data that they matter. The speculation that we focus on is that having memory CD4 T cells at time 0 that can recognize SARS-2 would be likely to facilitate a faster, better, stronger antibody response to SARS-2 than if you had weak or no T cells at time 0. That's just, that would be a classic memory response to help a new antibody response. So we would predict that having cross-reactive T cells would be a positive. It's also possible that they're a negative somehow, and so it is important to keep an eye out whether they have some pathogenic role. There is literature on this topic that we lean on from influenza, and so it's been known in past influenza pandemics or in influenza shifts that if people have pre-existing cross-reactive T cells, that can provide a protective benefit to a new flu, even in the absence of cross-reactive neutralizing antibodies. And so that is part of why we speculate that such a thing may be happening in people who have apparent cross-reactivity between SARS-2 and common cold coronaviruses, although again, it's speculation at this point that it's cross-reactive with common cold coronaviruses, and we are working to directly demonstrate that with the SETI lab. So this has been a highly collaborative project between my group and Alex Setti's group, really led by Alba Grifoni and Daniela Weisskopf, with Sydney Ramirez, a clinician in my group, and a whole bunch of other people working, including a lot of people at UCSD. This work was essentially entirely funded by the NIH, as well as a substantial contribution by an anonymous donor, which has been really helpful because this work has been really expensive, and we would be happy to get more support. So I will stop there and be happy to open up the Q&A led by Sabrina.
Thank you so much, Shane. That was great. We are getting a lot of questions. I'm just going to go back to sharing my screen and we can go through the Q&A. We've received a couple of questions about IgA responses. People are noticing high IgA responses to the SARS-2 spike. What is it doing? There seems to be conflicting information in the literature, and I think people were interested in the context of T-cell responses, what your thoughts were on IgA.
Yeah, great questions. So we've seen IgA, we've seen RBD and spike-specific IgA, as well as nucleocapsid IgA in the vast majority of people. There was a fantastic paper in Nature Medicine looking at a large group of individuals in China that the vast majority of people developed both IgG and IgA on similar kinetics, and actually IgM being less, which is somewhat counterintuitive. In the cohort I just showed, and in a follow-up cohort, we've seen that the IgA response correlates well with the spike-specific CD4 T-cell response. I would, certainly based on first principles, one would expect IgA to be valuable in protection against a respiratory infection like SARS-CoV-2. It is important to note that in lung, IgG does get transidated into lung quite well. And so even in the absence of an IgA response, one would expect IgG to do well at stopping or slowing a viral infection. So to me, it's again, just based on first principles, what you want to focus on, what is most likely to be relevant is the combination of the IgG plus IgA neutralizing antibody titer, as both are likely to be present in lung and nasopharynx and contribute. And if you just look at one or the other, there is variability between people. Some people have more of one than the other, and just the guess would be both would be relevant for protective immunity. And that's just coming from other respiratory viral infections. There's no direct evidence of that for SARS-CoV-2.
Okay, great. We received a few questions about the AIM assay. What are your thoughts about the use of the AIM assay for studying antigen-specific T follicular helper cells in mice? Are the induced activation markers across NHP, human, and mice conserved? There were a couple of questions about AIM. If you can comment on that, please.
Yeah, that's a great question. So my lab does mouse work, monkey work, and human work. And just for historical reasons, we initially didn't need an AIM assay in mice because we had other ways of tracking the antigen-specific TFH cells, where it was really in humans and monkeys that we were forced to reckon with the fact that that GCTFH were essentially undetectable with cytokine assays in the immunizations that we were studying. So we have now gone back and done mouse AIM assay development. The markers are not all the same. For example, initially in humans, we actually quite like CD25 as an AIM marker in GCTFH, but in mice that's not acceptable because all Tregs in mice express very high levels of CD25, and so it becomes a mess. We have a manuscript submitted on that now, and actually we should post it online. And another group has published something similar, but we use for mouse we like surface CD40 ligand and CD69 actually, and just a short 6-hour assay is working quite nicely for us for antigen-specific TFH cells, as well as other antigen-specific cells. For humans and monkeys, we find that for GCTFH, those markers actually don't work particularly well because in humans and monkeys, there's already so much CD69 and variable surface CD40 ligand that we have to choose other markers in primates and humans. But in mice, surface CD40 ligand and CD69 work nicely. And just to detect the surface CD40 ligand, you have to block CD40. Yeah, and there's a paper on that, or I can send you a copy of our manuscript if anybody wants to email me.
That'd be great. Okay, we received a few questions on the cohorts of the population involved in your study. You could just comment on the age of subjects that were part of the pooled T-cell analysis and if they were hospitalized versus not hospitalized. And yeah, comment on the study population, please.
Yeah, great question. Sorry if I wasn't sufficiently clear about that. So when I say average cases of COVID-19. So I said convalescent. So these were right. So these are post-infection cases, and by average I mean non-hospitalized. None of those people were hospitalized. All of them were PCR confirmed. We didn't allow anybody in the study who was not a PCR confirmed case, and everybody was symptomatic. The symptoms ranged, as has been the case for more broadly, but frequently, shortness of breath, weakness, dry cough, some GI symptoms or anosmia, but no hospitalization. So all of that was non-hospitalized cases again, which we infer to be your average COVID-19 case and representative of what a normal, successful immune response to it is. The people ranged, I don't have the number right in front of me, either late 20s or 30, up to I think 65-ish in that initial cohort. And whereas most other groups have focused much more so on hospitalized cases and really ICU cases,
Sure. Yeah.
In part because those are the patients that people have had access to, right? They're in front of them. The challenge with those is that frequently, well, those are relatively extreme cases, right? So they're important cases, but they're relatively extreme. And frequently, those people are being studied once they're already severely ill. And so a challenge there becomes, once one tries to look at the immune response in those individuals, it's very hard to tell what went wrong when in chicken and egg type problems. For example, if we did similar studies in a mouse model looking at progression of severe disease, if we looked at the time the mice were already really sick, we would actually make a different conclusion about the importance of the immune response than if we looked earlier when actually the key factors of the immune response were happening. And so I think those are the challenges with studying hospitalized cases. But that's what we're doing now. We've got a cohort of hospitalized cases, acute cases, hospitalized cases, and convalescent, and now we're cross-referencing the antigen-specific CD4, CD8, and neutralizing antibody responses across those to try and get a sense of is severe acute disease associated with problems in adaptive immune responses, or is severe acute disease fundamentally about non-immunological risk factors such as cardiovascular disease or other risk factors that people have previously reported on, because I think that's both important for clinical management of the disease as well as understanding what a vaccine might need to do, right, to protect people at highest risk.
Absolutely. That kind of dovetails nicely into a couple of questions we were getting about vaccine efficacy. So in aging adults, they're specifically the most vulnerable to COVID-19 so far. Their response may be limited by immune senescence, comorbidities. Have you been able to assess adaptive immune responses in aging adults? And if there are differences between young and aging adults, have those been observed?
Yeah, great question, and definitely an important question. And yeah, we're doing that right now. We have patients, I think, up to age 85, and we're trying to enroll more people as well to directly assess how much, if any, difference is there in the adaptive immune response between young and elderly, or is susceptibility of elderly due to other issues of frailty that are non-immunological. So those are critical questions, and we're working on them, and hopefully other people are working on them. And we're also happy to share reagents or protocols with any groups that are working on those questions. We're happy to share any of our materials that we have pre-publication, or if people, one of the challenges with these is when you want to ask questions, as was just described about elderly versus younger people, it's important to get a large enough cohort, right, that you can make conclusions and show. We've initiated collaborations to do that, and we're happy to initiate more collaborations to get enough samples to understand those outcomes.
Great. Yeah, and your lab famously has been involved in some of the original work around lymph node fine needle aspirate research. We're getting a couple of questions about sampling in relation to that. In the CoV-2 and CoV-2 negative studies, have you been sampling PBMC T cells, or have you been able to do fine needle aspirates to get germinal center cells from lymph nodes?
Yeah, so all the data that was just shown was conventional PBMCs. We don't expect, in the context of infected people, that lymph node fine needle aspirates would be likely to be particularly productive, because we would expect most of the activity in the lymph nodes to be in lung draining lymph nodes, which are far too deep to access with a conventional lymph node fine needle aspirate. However, in the context of immunizations, I absolutely think lymph node fine needle aspirates could be very valuable in vaccine trials, because we've seen in preclinical studies in monkeys and in unpublished human clinical trial studies led by HV10 and others, building on our lymph node FNA work, that lymph node FNAs have been very informative for germinal center responses and other immune responses that may not be measured or reflected in blood, and one can get the lymph node fine needle aspirate data quite quickly, 3 weeks after the first immunization, that possibly is predictive of outcomes 6 months later. So by directly looking in the lymph nodes of an immunized person, or most of what we've done, immunized monkeys. When you have a vaccine candidate where you're trying to get neutralizing antibody responses, first priming responses, frequently aren't particularly strong in the blood, but there can be a ton of activity going on in the lymph node. And so by directly sampling the lymph node, I think you can get a better prediction of how high quality that immune response might be at the end of your immunization regimen, which may be 6 months later.
Okay, we just got a couple more minutes for questions. Before we ask additional questions, we've received a lot of questions, Shane. I'll send the rest of them to you at the end. So you can take a look. I'm sure you have time to answer the 50 questions we've received in the chat. Let's see. Do you have any final thoughts that you wanted to share before I ask you just a couple more questions? Anything, in terms of next steps or the next study you'd really like to see done?
Say that again.
Just in relation to vaccine development and next steps for your work, can you share a little bit more about what the next steps would be and the next studies coming up in your lab?
Yeah, sure. I mean, I think broadly speaking, obviously the advancements in immunology and vaccine work with COVID-19 globally have been phenomenal. Just the speed at which people have worked and been creative and collaborated and got things published has been really quite extraordinary.
Absolutely.
On the vaccine development side, which is absolutely critical, most of the published work so far, I would say, is encouraging in terms of the protection studies in monkeys that have been reported, that people have been able to protect monkeys from disease with first generation vaccine efforts. I find that very encouraging. Due to time constraints, right, durability certainly has not been demonstrated for any vaccine candidates, but at least being able to get protection at all from disease, clinical disease, I think has been very, very encouraging. I think the negatives have been the neutralizing antibody titers, and essentially all of those studies have been low. And so I think it's plausible that other vaccine strategies maybe need to be employed to get better neutralizing antibody responses, and that may relate to paying more attention to the T cell response, right? The TFH and the CD4s that help drive those B cell responses. And I also think it would be wise to just consider the possibility that while neutralizing antibodies are great and wonderful, that it may be that CD4s and CD8s play as big or bigger role in normal control of COVID-19. And so vaccine efforts that at least explore those possibilities would be wise.
Relatedly, we're getting some questions about the antibody response and the durability of antibodies. A few people asked whether people who had severe disease would have more durable antibodies or a better protective response. And a few folks have asked about the durability of antibodies in convalescent serum. So could you just go into a little detail about durability of antibodies and responses in different categories of the populations you've looked at?
Yeah, so those are great questions, and a lot of people are wondering that and studying that. And there was I think a different paper in Nature Medicine, right, recently that came out on durability, and I think there's some preprints on the topic as well. The only way to measure durability is to wait, and so right now everything's still pretty short term as it relates to durability. There are absolutely concerns about antibody response durability. post-infection. And there's essentially no data on T-cell durability post-infection, i.e., taking multiple time points post-infection and observing longitudinally, is the memory stable or does it decline? Just based on other situations, it's also not clear how long one needs to measure those responses to know whether durability is an issue or not. And what I mean by that is there are certainly scenarios where post-infection or post-immunization, there's a drop-off in responses over a few months and then a stable phase. But then there are also scenarios of just continuous drop-off. Those aspects of memory like that, kinetics and durability of memory are in humans are not predictable, essentially, at this point. There's not enough immunological know-how to do that. So I think everyone is right to be concerned, and the data just has to keep coming in to get a sense of how big of an issue is that. Is there going to be a short-term decline and then stability, or really these responses, are they going to vanish? Even if the antibody responses disappear, do people maintain memory B cells, which in some other scenarios have been more durable and still may be valuable for protection? And same thing on the T cell side. Are the T cells, are the memory T cells durable, and are they associated with protective immunity? My normal statement on this general topic is, while we don't know what rules define immunological memory in humans, immunological memory in humans largely seems to track with the severity of the initial insult, much the way human brain memory works.
Sure.
If you almost got hit by a truck and missed it, and survived, you're much likely to still remember that, several decades later, as opposed to, what color socks did you put on yesterday? You might not remember that, and that's because of the difference in the importance of the event. Your immune system certainly to some degree works in that same way. We showed, for example, with the smallpox vaccine, when I did my postdoctoral work with Rafi Ahmed, that a single immunization with the smallpox vaccine could result in circulating memory B cells 50, 60 years later in a person. So it's certainly possible for people to have immunological memory to a single exposure 50, 60 years later. And Jim Crow, who spoke on a previous human vaccine project talk, right, he did something similar subsequently with 1918 flu. He found, I think, 80-year-olds who, still had memory B cells to that infection. And so it's definitely plausible that there may be a range of outcomes with this particular SARS-CoV-2.
Definitely. We're about at the end of the hour. There was one question I thought was interesting about vaccine safety, in particular immunopotentiation. It's a big issue potentially for SARS-CoV-2 vaccines. What immunological readouts do you recommend as the best to predict the potential in both NHP and in vaccine clinical trials?
Yeah, I think that's a great question. I would generally expect mice to not be predictive in that regard. So I agree with the phrasing of the question of, what should you look at in monkeys or humans in those. I think looking at the qualities of the CD4 response are the best metrics to look at for that. I think biggest concerns from immunopotentiation come from whether an aberrant CD4 T cell response would be generated that might be highly polarized to Th2 or highly polarized to Th17 and not polarized to TFH or Th1, which would be the normal expectations for antiviral immunity, and matching what we're seeing in these COVID-19 patients, that the CD4s in those patients are largely Th1, and now some reports of CD4-CTL, Th1, CD4-CTL, and TFH. That's what you want to see. If you see other CD4s, well, sort of unpolarized CD4s is fine, like just single producers of IL-2 or CD40 ligand. But skewing in other directions, I think, would be cause for concern. I don't think serologically there's anything that I would consider predictive in terms of, in humans, an IgG1 response is expected to dominate, and an IgA response, I think, is a positive thing. So those would, again, those showing up would largely match what we're seeing in average COVID-19 cases, which I would correlate with positive protective immune responses.
Great, that's really helpful. All right, so we're over time actually. Shane, thank you so much for your time. I know you're incredibly busy and you're doing so much great work. We really appreciate your time and joining us today for this chat. Thank you so much.
And I've reached the bottom of my tea, so clearly.
Oh, perfect. Works out. And thanks to all of our attendees as well. The schedules are crazy these days. We're so glad you're able to join in these discussions. And I'll pass along the questions to Shane afterwards so that he can see what types of questions everyone is asking that we didn't reach.
Sure, and feel free to email me about collaborations or peptide pools if you want them from the SETI lab, or if you want to share samples with us, or if you need protocols for us. Really, we're very happy to share and help. So bye for now. Sabrina, thanks to the Human Vaccines Project for hosting.
Thank you so much. And our next session will be July 16th. I invite you to join us. It'll be at 10 a.m. Eastern Daylight Time. Our speakers will be Dr. Thomas Rogers from the Scripps Research Institute and Dr. Joe Jardine from the International AIDS Vaccine Initiative, who will present on a recent paper describing the rapid isolation of potent SARS-CoV-2 neutralizing antibodies and protection in a small animal model. And if you're interested in more research on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And please feel free to visit our website and follow us on LinkedIn. We'll upload a recording of this webinar as soon as possible on those sites. And have a great day, stay safe, and we hope you'll join us on the next Global COVID Lab meeting. Thanks, everybody.